I was asked to give a talk on what you can read here. So basically, that's a carte blanche, more or less. Here are my disclosures, not relevant for this talk. So basically, this is the task, and I said sure, and then I realized what have I done? As the standard book on imaging and oncology is about maybe one volume is about 5 centimeters thick, and there's a lot of stuff to imaging cancer, biology, etc. So maybe let me start quickly with what I think are the components of modern oncology / oncologic imaging. So you've got an image there, which is a PET scan, which is built on some underlying biology. Then you also can use, in addition, and I'm looking at the entire diagnosis here, in vitro diagnostics, so that is in this case microfluidics, but also gene, whole tumor sequencing, and things like that. Then there is modern novel imaging, and we're going to talk about that. Then there are probes. There's not a new agent in this case, but this is the contrast agent side if you want. And then you can top everything with artificial intelligence and machine learning. And that's pretty much what is being integrated currently into modern imaging. Now here are all the imaging modalities based on a paper from Feuchtslater in Boston. And I extended it with some additional modalities here. So PET MRI, and there's no spectral CT, intraoperative fluorescence reflectance imaging. Then there is multispectral optoacoustic tomography, so optoacoustic imaging. There is a high resolution version of that. And then here are other developments which are sort of preclinical. This is clinical, and we have MSOT and RSOM also preclinical. Here's high resolution microscopy, STORM, and successors, and then there are emerging modalities where PET is merged with high resolution ultrasound, for example. So there's a whole realm to play in. This has been shown in similar fashion. These are the different modalities. For the interest of time, I jumped over that. You probably have seen that quite a bit, maybe pointing out optical imaging goes nicely into the molecular realm. The resolution, if you look macroscopically, a couple millimeters or below that, of course, and then if you go to microscopy or way beyond that. So this is our clinical routine in cancer imaging, more or less. So this is molecular targeting with different radio tracers. Now we are talking nuclear imaging. And just to give you a taste of what we can do and see. So here we can look for bone remodeling. This is the bone scan. This is a FDG PET scan. Here are a lot of lesions in the pancreas and the patient with prostate cancer. We can image androgen receptor with specific ligands. This is androgen receptor. And this is looking at PSMA, prostate-specific membrane antigen. And you can imagine this is except that, but it could be these three are all in the same patient. So you get a total, literally different picture of a lot of lesions here with FDHT, quite fewer. With FDG, the bone scan is more or less non-specific. There are some lesions here, of course. And then you can go even more specifically into PSMA. With specific membrane antigen imaging. That basically means with molecular imaging, nuclear imaging, we are imaging molecular signatures. So here's an example from a patient which was imaged with GRPR2 with a gallium scan after 60 minutes, and you see some uptake here in the prostate, and you see here on G2 signal decrease, which denotes the tumor, also indicated by the decreased diffusion image on the MRI here. And I put these here because Jan has asked me general cancer imaging. So this would be, this is a little bit more out there, but T2, diffusion imaging, and some sort of PET imaging is certainly what we're doing routinely today. Here's another example, a PET positive case with a PSMA tracer. And here's also a prostate cancer patient, both have a Gleason 8 or 9, relatively aggressive tumor. This scan was negative. And there was very little PSMA staining in here and a lot of PSMA staining in there. And from the biology, you can deduct certain molecular signatures, for example, that in this tumor likely there was a much higher PI3 kinase mTOR signaling than in this one, and that might have therapy and surveillance consequences. Now, if you look at molecular medicine, this is supposed, it's not the actual chip, but it's sort of standing in for the impact chip, which is targeting 368 and I think it's now more actionable targets in the genome to look at mutations which can be acted upon in patients. Now, impact has these 368 plus targets, and if you look at the PSMA PET, you got one target. So we were basically running in imaging against molecular medicine by only being able to image one target at a time. And here you look at this whole range of multiple targets, probably more by now because the slide is maybe 2-3 years old now. Now, what is really needed in tumor imaging? I think one of the big parts is we want to image heterogeneity. We want to image the tumor microenvironment. That consists of multiple players. We need multispectral capabilities because that can really carry this multiple information within one scan, because you don't want a patient who is already sort of hit hard by the diagnosis to come back on one day for scan 1, on another day for scan 2, on another day for scan 3, and another day for scan 4, because time is valuable for these patients. So we want to include, interrogate tumor heterogeneity. As I said, the tumor microenvironment is important, and the tumor microenvironment are immune players, and there is something which is called, another the slide in here, pseudo-progression in modern immunotherapy, when the tumor initially increases in size and then it decreases. And if you just do a CT scan, it's a tough time saying, well, this therapy didn't work because the tumor progressed, or the therapy did work and it's pseudo-progression. The pseudo-progression is coming from an inflow of immune cells into the tumor, which basically adds cells, volume to the tumor, and it gets bigger. If we were able to show that these immune cells are going into the tumor, we would be able to tell much earlier that this is pseudo-progression and it's actually working rather than real progression. And you solve it currently by doing multiple scans within a time frame, and then over a couple months you see it's getting smaller, and if you see over a couple months, well, it's getting bigger, then the therapy didn't work. But you want to tell the patient that earlier and switch. There are other multi-parameter dimensions. Tumor microbiome, the gut microbiome, for example, would be very important to look at in my mind. And this is going towards precision medicine. And I think currently, at least, really multispectral, people are working, including us, and other options there. You can do multispectral with optical imaging. So just to show the fifth dimension of the tumor, so you've got tumor size and volume, that's 3D, that goes over time is your fourth dimension, and then the tumor complexity and heterogeneity really is a fifth, if not multiple more dimensions. And then changes in that tumor complexities, and yet again another dimension. And then you want to look at the stroma. In pancreas cancer, it's very stroma-rich, for example. As I mentioned, the immune cells, lymphatics play a role. So there are multiple things we want to interrogate. And this can change in different lesions, as was shown in the seminal paper, where they took the primary tumor and then metastases and sequenced the out of it. Excuse my French. They looked at multiple gene signatures and found that in between one patient, these change within the lesions. Currently, we can't image this. This was done post-mortem. Here's an example for, just to show you why it's important. This is Vimentin intensity in sort of a microscopy slide where they stained for Vimentin and then went really in and showed the expression levels in different cells. And you can see the range from low to high within the tumor itself. And this is just, of course, one signature of the Vimentin. And this was published in Nature Biomedical Engineering. Here's again the tumor microenvironment. You can see all these various players, which are all important. Initially we're just looking at tumors, and then we started to realize where there's more. I mentioned the immunotherapy, which are the T lymphocytes. People are now recognizing more that macrophages and NK cells are important. Vascularity is important, but post-dendritic cells have a role. So currently with FDG, we can image the tumor. And we can image the immune cells, but we can't separate them. It's all the same signal. We can use antibodies, which can show us CD8 cells, T cells. We can use Herceptin, that shows us only HER2-positive tumor cells in this case. MRI, ultrasound, and CT just lumps everything together at the moment. With optical, you can do more, but the penetration into the body is slow. You can't image something deep in the brain, or you can't image something deep in the liver. Optoacoustic imaging, as we are calling it, or photoacoustic, is multispectral and has some better penetration. So there's some good reason for working on that. So I work on that. So lasers, because this is Laserlab, so that's why I threw this slide in here. Therapeutics should read applications there, of course, for anything with higher energy lasers. We can do optical imaging, but I assume lasers are really getting more replaced by LEDs. You can see what the various applications are there. So we have the LEDs. Now, I just gonna go through some examples. Some are from us, some are from others. Here's the seminal paper where they used really the first intraoperative optical imaging. So they had a folate targeted agent. It was a multi-collaborative effort. And here's really the interesting part is that you see the white light, which the surgeon would see, and here's the fluorescent image, and you see all these peritoneal nets in this ovarian cancer patient. And they counted the number of lesions detectable just on the color image, which is this one, and with the aid of fluorescence imaging. And the fluorescence imaging really showed quite a significant higher number of lesions. And this is important for debulking surgery, where they try to remove as much peritoneal disease as possible. Because, of course, every little lesion left behind will give rise to recurrence. Here's an example from the former Rainer lab, Novartis, where they're looking at PARP imaging. PARP is involved in DNA repairs highly expressed in tumors, also the lymphocytes. But here they show PARP imaging in the tumor, and here's the border of the tumor, normal tissue, and here's the tumor tissue in an oral squamous cell carcinoma, and that led them to a phase 1 clinical trial to detect oral cancer with basically a mouthwash containing the fluorescent agent, and then the dentist or oral surgeon can look into the mouth and see the lesion there. And here's an example from this human phase 1, where you see in the fluorescence image tumor tissue here and non-tumor there. Here's the white light, and here's another angle. So this would be an example where you can do an easy detection of oral cancers. This was particularly useful in a study they did in India, where they were, I forgot what it was, they were chewing, I think it was betel nut or something, a drug-like thing, which causes a fibrosis in the mouth, and it can be quite debilitating. But it also causes squamous cell carcinomas, and it's very difficult to visually see the difference between the fibrotic tissue and the tumor tissue. And with this fluorescent agent, they were able to show in patients where the tumors are, and to basically elect patients which should undergo surgery. Now, I'm not sure if this has been mentioned before or missed it. The SWIR or near-infrared 2 window is going increasingly into focus. In the conventional near-infrared, up to 800, we have high absorption, high scattering, millimeter imaging depth. There's, of course, the near-infrared window where the water absorption is very low around here. And then now there's the shortwave infrared or NIR2 window, which goes from 900 roughly to 1700. There's almost no absorption there. It's low scattering, and you can go much deeper. And here's one of the first examples where they constructed an agent bound to, I think these were carbon nanotubes. And this is the same mouse image at normal infrared and here at near infrared, and you can see the penetration is much better, much less scatter diffusion, and you see the vessels much clearer. Here it's been used clinically with cetuximab iodide 800 on a patient, again with the oral carcinoma image, the base of mouth carcinoma image through the skin, and you can see the tumor here and even the process centrally in the tumor. Now, I can give a talk about optical imaging without at least throwing a couple of slides in there. This has nothing to do with lasers, but it's something we're working on, which is Cerenkov Luminescence, which is basically optical imaging of radio tracers. This is blue light, which you see in nuclear power plants. And what happens is when a charged particle travels through a dielectric medium, so that's tissue, water, faster than the speed of light, it emits a continuous spectrum of blue light, but it goes down into the visible and even further. This is an example of a positron going to a tank of water from Brian Coke, now in Alabama, Pittsburgh actually. And you see the shower of photons coming off there. This has been used clinically by surgeons with this device from LightPoint, which is basically a very sensitive because the light intensity is very low camera, and they're looking here at the breast tumor resection imaged with FDG, I think, and you can see the tumor giving Cerenkov light, and they were looking at the margins, whether they were clear, and in this case they were. And here's a lymph node which showed individual specks, and these were subcapsular tumor nests. And here's a prostate cancer choline specimen, and again, they were looking at the margin free. So this is ex vivo back table imaging with Cerenkov. We constructed a Cerenkov imaging suite to image patients entirely. This is now coming out relatively soon, we hope. In the journal mentioned there. And here are just 2 or 3 quick examples. This is a patient which underwent iodine-131 therapy. Here's the Cerenkov from this remnant in the thyroid bed. And here is an example of EPG of a patient with follicular lymphoma. You can see here the Cerenkov light coming from there, and here is gallium dotatate. These are lesions in the body, which we can see the spleen in the pancreatic lesion. And before I go over to the optoacoustic one, we think this is sort of not a cheap man's pet, but a way to do a quick nuclear scan, particularly in areas which have the ability to get radio tracers or don't have that many PET scanners around, like developing countries, India, South America, where you can get the tracer and do a quick, cheaper scan, sort of to triage patients for the next step. Now, let me go before I close, because I know it's late, to what we did in optoacoustic, which is sort of the competitor's talk, if you want, because this is a different company. They built an MSOP scanner, multispectral optoacoustic tomography, which has millimeter resolution. There's also a Raster scan optoacoustic mesoscopy scanner, which is a micrometer resolution. And we were lucky enough to get a prototype from Basilis and Christus in Munich into our lab to look at tumor biology. And what you see here is a subcutaneously implanted tumor, and we are looking at, I think it's 532 laser. So we're looking just at hemoglobin absorption, which gives us basically the vessels. And there are larger vessels which are captured in this frequency from the ultrasound, and smaller vessels, and then it's merged, which gives you this orange-yellow color. And we're looking at tumor vascularity. We now have a multispectral version of this. First, it was also a prototype. Now it's a commercial one, where we can look at oxy and deoxy hemoglobin at this resolution, which is very close to intravitreal microscopy almost. And then there's a clinical version, both of the lower and the higher resolution scanners, and we're starting clinical trial with MSOP equivalent clinically soon. But I want to go quickly into this paper here, where we looked at tumor vascularity at this high resolution. Here you can see a tumor growing. We can get 3D MIPS of this, and we use this to interrogate a vascular-targeted photodynamic therapy, which is coming out of the Weizmann Institute in Israel. It's called WSD11. It's based on chlorophyll. It's a vascular-targeted agent, and what it does, it upon irradiation in the near infrared, it basically obliterates through a known mechanism the vessels. There are radical oxygen species are created, and they destroy the vessels, and they destroy them pretty quickly. So here's a tumor image 3. The agent was injected, laser light on, and after 5 minutes, you can see that this vessel starts to getting thinner. Here it's already disrupted, here it's gone, and then after an hour, you see how this is getting pretty chaotic, and these nicely organized vessels are disappearing. And if you measure the vessel fragments over time, you see that you get an increase of vessel fragments. You can sort of quench the process with hydroxycobalamin, which is a nitric oxygen radical scavenger, and then you don't see any effects and you don't see any vessel fragments. Now, at later time points, at 1 day, it's interesting. At 2 days and 3 days, 5 days, you see how the tumor is basically falling apart. There are no vessel structures. And all you see here is hemorrhage, more or less. You can see that in the darkness of the tumor. Interestingly, at day 1, you see a nice, very pretty vascular tree. It's sort of what Tess Jane was always promoting, the normalization of the vasculature upon therapy. We did an experiment also back then in Munich looking at oxy and deoxyhemoglobin with another prototype scanner. And this is just a general oxygenation map pre and post, and you can see how the vascularization and the oxygenation is much better, and it now falls apart to the lower oxygen area after this therapy. And this is shown again. The interesting part, which excited our Israeli colleagues, was that after 5 minutes, you get sort of an oxygen burst. And they were excited because you need the oxygen actually, of course, for the therapy to work, because it creates radical oxygen species, and you need oxygen for that. So that's first to get a burst, and then you see how the oxygenation clearly goes down. And I think this is the second to last slide. So these were the guys working on that. These are 2 different urothelial tumors, because Jonathan at Sloan is a urologist. And this guy, this tumor is responsive to the therapy, while the UMOP3 is resistant. And if you look at the optoacoustic images, first of all, it's not a black and white sort of picture you see. You see that the vessels are being destroyed. After 3 days, it looks pretty bad, but you start to see then on this guy after 7 days already, and after 14 days, that vascularization is coming back from the rim. And these are starting to regrow and reoccur. If you look at staining at 14 days, there's basically very little left. There's a lot of necrosis, and you see nice vascularity in tumors. And we looked at parameters which are able to predict the recurrence, and if you do the large vessel fraction over the total vascular area, you can see that at day 3, it got bigger on the 567, which is basically the responding. So the vessels are falling apart until they don't. So at day 3, you can already sort of predict what is happening. You can't do that from the growth curves. They are not different at this time point. We developed some agents just to show that quickly. Quenchers is contrast agents for optoacoustic. Here's a black hole quencher 1, imaging RGB, so the vessels, and then again from Tom Reiner's lab when it was still at Sloan. There's a whole library of quenchers we went through and checked which one works best for optoacoustic imaging as contrast agent here, the images. And with that, I'm done. And if you've got any questions, maybe you can send me an email. Thank you.